Hello everyone. So in this video, we are going to talk about test tube baby. What is a test tube baby? Can a baby be produced in a laboratory? Why do we call it as a test tube baby? Let us discuss all these points. So very first thing is, what is a test tube baby? Test tube baby is not a baby which is produced in a laboratory. In fact, it is a part of a process. It's a part of a process in which the childbirth, in which the process of fertilization is done in laboratory. Okay, the process of fertilization, only one single part of the reproduction is done in the laboratory. In 1934, first time a rabbit was produced by in vitro fertilization. In vitro fertilization means it is a process in which the fertilization is done in laboratory, in vitreous apparatus, that is in a glass or in a test tube or in a petri dish or something like that. In 1978, the world's first baby was conceived, human baby was conceived by in vitro fertilization in Manchester, UK, and the baby was born and her name was Louise Brown. As the medicine advanced, IVF, that is in vitro fertilization, was transformed into a natural research to stimulate a clinical treatment. So in this, actually, what we are doing, that is, we are creating a condition similar to that of the body condition, and making the sperm and egg fertilized or meet together and fertilize. So there are several cases when, due to some of the defect or abnormalities, the couple has to go for it. So what are the reasons? Like if there is a blockade in the reproductive canals of the female, or the sperms and of the male is not compatible with the female reproductive system, or when they are not, the female is unable to produce egg, or when the sperm is unable to fertilize the egg normally due to some reason, or when the sperm count is poor, that is the number of sperm which are required in order to carry out fertilization is not enough, or when the male partner is not nearby, he is somewhere away or may not be reaching into this thing. Suppose someone is gone to the space mission, or someone is gone to Antarctica mission, or someone is on the border, or someone is paralyzed and he or she is not he is not able to carry out the fertilization, then the only way is to remove the semen and can fertilize it in the laboratory. So first egg is collected from the female partner, and normally a female partner produces only one egg in per menstrual cycle, but here we are giving some special hormones to get more than one egg. This can be done either collecting the eggs which are produced in the different menstrual cycle and cryopreserving them, or by giving the special treatment of the hormones. So there are a large number of eggs collected, generally 2 or 3, then they are kept in the petri dish. Sperms are collected from the male and the fertilization is done in the laboratory, and then soon the zygote will start dividing and it reaches to 8 cell stage. Then it is transferred to the fallopian tube of the female, and if it is more than 8 cells, that is 16 or more cells, then it is transplanted into the uterus. Okay, so the entire process is summarized here. First, the hormonal injections are given, the eggs are collected, the sperms are collected from the male, both are brought to the laboratory, the fertilization is done, the cell division begins. When 8 cell stage is reached, they are to be transplanted into fallopian tube. If more than 8 cells, then they are to be transplanted into the uterus, and further development take place in the mother. And when the child is completing its time, then she is delivered. So entire process is summarized here. That's all in this video. Thank you.